High-speed chemical reactions in microdroplets represent a fascinating frontier in reaction kinetics research. In these tiny droplets, often with volumes in the pico to nanoliter range, the confined environment fundamentally alters the behavior of chemical reactions. Unlike bulk solutions, where molecules have ample space to diffuse and interact, microdroplets force reactants into close proximity. This confinement can lead to significantly higher local concentrations and enhanced collision rates, accelerating reaction speeds beyond what is observed in conventional setups. One key factor driving this acceleration is the high surface-to-volume ratio inherent in microdroplets. The droplet interface plays an active role in the reaction by providing unique sites for adsorption and reaction. For example, molecules may preferentially accumulate at the interface, where different physical properties, such as polarity and dielectric constant, can lower the activation energy barrier. Have you ever wondered how a reaction that takes hours in a flask can occur in mere milliseconds in a microdroplet? The answer is that the confined space and active interfaces create conditions that are not just scaled-down versions of bulk processes, but entirely new environments where reaction pathways can be altered or enhanced. In addition to the surface effects, rapid solvent evaporation in microdroplets can further concentrate reactants, leading to conditions that mimic high-pressure environments. This transient concentration effect often results in reaction rates that are orders of magnitude faster than in bulk solution. Researchers have demonstrated, for instance, that oxidation reactions or certain organic syntheses proceed remarkably quickly when performed in microdroplet form. By controlling the droplet size and the surrounding atmosphere, scientists can even fine-tune the reaction conditions to achieve optimal yields and selectivities that are difficult or impossible to replicate in traditional reactors. Another intriguing aspect of microdroplet chemistry is the way it enables the study of reaction mechanisms under extreme confinement. The short timescales involved, from microseconds to milliseconds, allow researchers to capture transient intermediates that would otherwise be too fleeting to observe. This opens up new avenues for understanding complex reaction pathways and developing novel catalysts. For example, when exploring enzyme-catalyzed reactions in microdroplets, scientists have found that the altered microenvironment can lead to enhanced enzyme activity or even reveal alternative catalytic routes. How does this change our understanding of biocatalysis? It challenges the traditional view by demonstrating that reaction environments play a crucial role in dictating enzyme behavior, potentially guiding the design of more efficient biocatalytic systems. Unexpected applications of these findings are already emerging in pharmaceutical research. The ability to conduct high-speed reactions in microdroplets allows for rapid synthesis and screening of drug candidates, significantly shortening the development cycle. Environmental chemists are also leveraging microdroplet technology to model atmospheric reactions, helping to better understand processes such as aerosol formation and pollutant degradation. Could these techniques eventually lead to breakthroughs in climate science? The answer is yes. By replicating atmospheric conditions in a controlled, confined environment, researchers can gain insights into reaction kinetics that drive critical environmental processes. In summary, the confined environment of microdroplets creates unique conditions that accelerate chemical reactions through enhanced molecular collisions, surface-driven effects, and rapid concentration changes. This has opened up exciting new avenues for studying reaction kinetics, allowing scientists to observe and harness reaction pathways that are inaccessible in bulk systems. As research in this area continues to expand, microdroplet chemistry promises not only to revolutionize our understanding of fundamental chemical processes, but also to enable innovative applications across fields as diverse as drug discovery, environmental science, and materials synthesis. Microdroplets provide a unique reaction environment where surface effects, reduced diffusion distances, and enhanced reactant concentration accelerate chemical transformations. Unlike bulk phase reactions, microdroplet chemistry benefits from extreme confinement, leading to faster molecular interactions and higher effective reagent concentrations. This acceleration is often linked to surface-induced catalysis and microdroplet-specific electric fields that alter activation energies. Studies have shown that certain organic reactions, such as peptide bond formation and oxidation reactions, proceed up to 1 million times faster in microdroplets than in bulk solution. This is like cooking food in a pressure cooker instead of a regular pot. Higher localized pressure and temperature speed up the process dramatically. Microdroplets have a high surface area-to-volume ratio, exposing a significant fraction of molecules to interface effects that can modify reaction dynamics. Surface tension and molecular orientation at the air-liquid interface create unique reaction conditions not found in bulk solutions. Charged species and radicals often accumulate at the interface, altering reaction pathways and enabling previously unattainable transformation. Electrospray ionization mass spectrometry, ESI-MS, has revealed that oxidation and polymerization reactions occur preferentially at droplet surfaces, sometimes bypassing the need for external catalysts. This is like stirring a drink with a spoon. Molecules at the surface mix and interact much faster than those deep inside the liquid. Microdroplets act as nanoreactors, providing localized reaction conditions that facilitate catalysis at scales previously difficult to achieve. The confinement effect leads to higher local concentrations of catalysts and reactants, improving catalytic efficiency. Some reactions that are sluggish in bulk solutions proceed efficiently within microdroplets due to altered solvation and enhanced reagent availability. Enzymatic reactions have demonstrated significantly higher turnover rates in microdroplets, suggesting that biomolecular catalysis could be optimized using microfluidic droplets. This is like shrinking a chemical plant into a single droplet where all necessary reactants are packed together, making reactions happen much faster. In charged microdroplets, electrostatic effects can alter reaction kinetics by stabilizing transition states or promoting selective bond activation. Charged microdroplets generated via electrospray ionization create high local electric fields, lowering activation barriers and increasing reaction rates. These electric fields also facilitate ion-induced chemistry where charged intermediates drive novel reaction mechanisms. Protonation, chemical reactions such as acid-catalyzed hydrolysis proceed orders of magnitude faster in charged microdroplets compared to bulk phase conditions. This is like using a microwave instead of an oven. Electromagnetic effects accelerate heating and chemical changes at a fundamental level. Microdroplet reactions occur under non-equilibrium conditions where rapid solvent evaporation and interfacial effects shift reaction equilibria. These deviations from bulk equilibrium allow access to reaction pathways that are otherwise inaccessible, enabling high-yield synthesis of metastable or reactive intermediates. The rapid solvent evaporation can also lead to concentration gradients, favoring selective product formation. Studies have reported the formation of complex organic molecules in atmospheric microdroplets, providing potential insights into prebiotic chemistry and the origins of life. This is like flash freezing food. Rapid environmental changes preserve intermediate states that would otherwise decay under normal conditions. Gas phase reactants can dissolve rapidly into microdroplets, leading to enhanced reaction rates at liquid-gas interfaces. This effect is particularly useful in oxidative reactions where dissolved oxygen or other gaseous species directly interact with reactants within the droplet. The rapid diffusion of gas molecules within small liquid volumes further accelerates reaction kinetics. Carbon dioxide absorption into water microdroplets has been shown to drive enhanced carbonate and bicarbonate formation, with implications for atmospheric chemistry and carbon capture technologies. This is like aerating wine. Exposing it to oxygen quickly changes its chemical profile compared to letting it sit in a closed bottle. Microfluidic platforms allow precise control over microdroplet formation, enabling systematic studies of reaction kinetics under varying conditions. Droplet size, flow rate, and reagent concentration can be fine-tuned to optimize reaction speed and selectivity. Microfluidic chips also facilitate automated, high-throughput screening of reaction conditions, In a fraction of the time needed for traditional experiments, microfluidic-based synthesis of nanoparticles has demonstrated superior control over particle size and composition due to precise reaction confinement within droplets. This is like using a 3D printer for chemical reactions. Precise control over inputs leads to highly reproducible outputs. Microdroplets provide an environment where biomolecular interactions such as protein folding and DNA amplification occur with enhanced efficiency. The confined space and high surface-to-volume ratio lead to accelerated enzyme kinetics and improved reaction yields. These advantages make microdroplets particularly useful in single-cell analysis and molecular diagnostics. Microdroplet PCR polymerase chain reaction enables rapid DNA amplification, significantly reducing reaction time compared to traditional bulk phase PCR. This is like using a high-speed blender instead of stirring by hand. More efficient mixing leads to faster reactions. The ability to accelerate reactions in microdroplets opens new frontiers in chemical synthesis, drug discovery, and environmental monitoring. By combining microfluidics with real-time analytical techniques like mass spectrometry, researchers can explore reaction pathways with unprecedented speed and detail. Future developments may lead to fully automated, AI-driven chemical synthesis platforms that exploit microdroplet kinetics for on-demand molecule fabrication. Continuous flow microdroplet reactors are being explored for the rapid synthesis of pharmaceutical compounds, allowing for real-time drug production in hospital settings. This is like having an espresso machine for chemistry. High speed, high efficiency, synthesis on demand.